So we are going to start. We have a lot to cover today. If we don't get through everything today, then I have some time on Wednesday that we can finish covering this stuff. But we're going to start in with aging and dementia and stress's potential role in sort of speeding up the natural aging process, which can be a negative thing. And then we'll talk a little bit about sort of the latest things that we know about Alzheimer's disease. So we're going to start off. We haven't heard from Dr. Sapolsky in a while.
We're just taking fingerprints because no baboon has the same fingerprint as another one. So we just took Coney Bear's, and I'm hoping to go to Riff and get his.
During this year's multi-generational research, Robert, who has spent his career documenting stress's effects on the individual and on the cell, tracks the trail of stress even deeper into our bodies.
One of the most interesting new directions of stress research is taking the effects of stress down to a nuts and bolts level of how cells work, how genes work, that half a dozen years ago nobody could have imagined.
The once unimaginable genetic structures called telomeres, which protect the ends of our chromosomes from fraying. As we age, our telomeres shorten.
What's interesting is stress, by way of stress hormones, can accelerate the shortening of telomeres. So the assumption is for the exact same age guys, if you're a low-ranking guy who's just marinating in stress hormones, your telomeres are going to be shorter.
So how does this formidable finding apply to us? San Rafael, California. Once a week, Janet Lawson keeps a very important appointment. She joins other mothers who share circumstances that produce chronic, unremitting stress.
So she loses her balance, and that's the scary part. So we just went out and she, let's say, bought a new helmet just for fun. And as she's getting older and wanting more independence, it's getting harder.
Each of these women is mother to a disabled child.
And for us, my son's only 8, and there's enough I can handle, and I don't allow myself to go too much out. I can't.
I had a friend recently who said to me, you know, I think you really should consider putting Flexi in a home. And that was really stressful in and of itself to think, wow. So, sorry. So I was like, wow, how could you even say that? She's, you know, a girlfriend. She's, even though she can't really communicate, she loves. She loves. She loves.
These remarkable women came to the attention of biologist Dr. Elizabeth Blackburn.
I don't directly know the individuals, but I know the stories. I'm a mother myself, and so when I heard about this cohort, I really thought it was worthwhile finding out what really is happening at the heart of the cells in these mothers who are doing such a difficult thing for such a long time.
Dr. Blackburn is a leader in the field of telomere research.
We have 46 chromosomes, and they're capped off at each end by telomeres. Nobody knew in humans whether telomeres and their fraying down over life would be affected by chronic stress. And so we decided we would look at this cohort of chronically stressed mothers, and we decided to ask what's happening to their telomeres and to the maintenance of their telomeres. What we found was the length of the telomeres directly relates to the amount of stress somebody is under and the number of years that they've been under the stress.
Such stressed mothers became the focus of a study by Dr. Blackburn's colleague, psychologist Alyssa Eppel.
Mothers of young children are a highly stressed group. They're often balancing competing demands like work and child rearing, and often don't have time to take care of themselves. So if you add on top of that the extra burden of caring for a child with special needs, it can be overwhelming. It can tax the very reserves that sustain people, and if they're stressed, if they're poor stress, they tend to die earlier.
These women have shortened telomeres, decreased activity of this enzyme, and very, very rough number for every year you were taking care of a chronically ill child, you got roughly 6 years worth of aging.
This is real. This is not just somebody whining. This is real, medically serious aging going on, and we can see that it's actually caused by the chronic stress.
But there is hope. Dr. Blackburn co-discovered an enzyme, telomerase, that can repair the damage.
It's what I always call the threat of hope.
Preliminary data suggests that a meeting of minds such as this may actually have a health benefit by stimulating the healing effects of telomerase.
And laugh.
If you don't laugh, forget it. You can't handle it. It's
what I found is that humor is something, there's a certain level of black humor that we have about our kids that only we appreciate. We're the only ones who get the jokes. In a way, we're the only ones who are allowed to laugh at the jokes.
One of the questions in the stress field is, what are the active ingredients that reduce stress and that promote longevity and compassion and caring for others may be one of those most important ingredients. So those may be the factors that promote longevity and increase telomerase and keep our cells rejuvenating and regenerating.
So perhaps connecting with and helping others can help us to mend ourselves and maybe even live longer, healthier lives.
All right. So, I think it is important as we're, finishing up, going through these sections, and we can spend all this time talking about stress, to realize that yes, stress does have negative effects on almost every cell in our body, but there are things we can do to minimize stress. There's things that we can do to repair damage from stress. And part of that is having that neuroeducation and understanding how these processes work, right? Getting back into aging. Life expectancy has increased, and I'll just point out this little blip right here. That was the 1918 pandemic flu. And we've already experienced about a 1.5-year decrease in life expectancy over the last year from the effects of the pandemic this time around. So it won't be as big a drop as it was there, because not as much of the population is dying, because we have better medical care now than we did back then, and better treatments. But it is going to have an effect there. So we can directly relate increases here from in the early 1900s, the average life expectancy only being 50 years old, right? Or 50% of the population only living to be 50 years old, to now having 50% of the population living to be upwards towards 80 years old, right? So much. I think the average life expectancy, when I say average life expectancy, that's at 50% number. And I think it dropped this year from 75 to 73.5, something like that. All right, so as we have made medical advances to now live 25 years longer as a population, right? And that's half the population, so half the population is living much longer than 75. We're starting to see increased prevalence in age-related diseases like dementia and Alzheimer's disease that we just didn't have to deal with 100 years ago because people never lived long enough to see those effects, or effects of chronic cardiovascular health and these sorts of things, right? So, stress and aging. So when we talk about aging, what do we mean by aging? What is elderly? So we're going to define elderly as people that are 70 plus, that are at that sort of 50% life expectancy. Not people that are in their 60s or 50s, but people that are up in that 70 and older range. And we saw some research articles last week, or the week before, about how the stress system changes during aging. In particular, the brake system, the ability to turn off and regulate basal glucocorticoid levels, in some, not all, but some aging people, becomes weaker, right? And so we can kind of separate elderly people into those that have good systems for regulating and those that don't. Why do some not have good systems? We don't really know. It's complicated. It could be an accumulation of more stress across their lifespan. But those are difficult studies to do. What we do know is that in elderly, that brake system is weaker, and so these hormones, norepinephrine, glucocorticoid, epinephrine, stay circulating longer when they get stimulated in response to a physical or psychological stressor. And as I mentioned, there's a variety, right? So we can separate people out to low and high groups. In general, it tends to creep up, and so just like your brain is not as healthy when you're 70 as when you're 25, your stress system is not as resilient when you're 70 versus 25. We also, last week, saw some research talking about the relationship between a healthy hippocampus and glucocorticoid stress systems in aging. And we know that, right, that the hippocampus can accumulate insults over time with repeated exposure to those glucocorticoids. And all of that stems from, again, decreases in receptors, decreases in the ability to recognize circulating glucocorticoids, which then elevates sort of basal levels, maybe not too stressful levels, but they start creeping up. So Sapolsky talks in this chapter about a good animal model for this increase in glucocorticoids with aging, which is in salmon. And so salmon, after they spawn, for some reason, die a few weeks after spawning. And this is common across 5 different species of salmon. It's also found in some other animals. And nobody really knows why this happens, but they do know how it happens, right? So they don't know why salmon die after spawning. If it's part of population control, who knows? But when we look at the molecular changes that are occurring in salmon after spawning, it is a downregulation of the ability to control glucocorticoids, right? So something about the spawning process in salmon desensitizes their brain to glucocorticoids to the point that they essentially eliminate the negative feedback loop. So they eliminate the ability to turn off, to recognize high glucocorticoids and have that natural shutdown. And so then what happens, well, those levels become extremely elevated. The adrenal glands swell from overuse, and all the negative effects that we know happen with chronic, prolonged, very high levels of glucocorticoids occur. So they get ulcers, they get kidney problems, the amino system gets suppressed to the point that they become infected with parasites and bacteria and die from it. Right? So so studies have been done where they remove the adrenal gland after spawning, and these salmon live another year or so. So it clearly is this having the stress system go off the rails, and if we eliminate the ability to secrete cortisol, they don't have this programmed cell death. It's almost like a programmed cell death. It's complicated programmed cell death through the release of these glucocorticoids. All right, so in talking about what happens to our brain as we age. As we age, we're going to lose neurons, and that's normal. And so I mentioned before that if you're fortunate enough to live to be 85 years old, you're not going to get around as quickly. Your motor system's not going to work as well. And part of that is your cortical motor system, not just your muscles and joints. Your cognition will slow, right? Your memory won't be as good in general. And this is called senile dementia. It's a normal thing. It's not an abnormal thing. And we think this is because of limited resources. We get an accumulation of insults. Those telomeres that they talked about are decreasing, and so our DNA becomes corrupted, and we're not able to replicate cells in the right way. And some people have suggested there's just simply an aging program. And a lot of this work has looked across species, and there's some species like tortoises that live to be 150 years old, and looking at, okay, how does aging happen in their bodies versus a mouse that only lives to be 2 years old, right? And that's normal aging, and what are the different metabolic rates, and what are the different changes that are happening. It looks like there's just, for each species, sort of a lifespan of it doesn't matter how good your care is, everything sort of, you have this programmed end of cells at some point. All right, so in looking at abnormal cognitive function as we age, the DSM-5 has a pretty complicated way of breaking this down in the neuro DSM-5. So they have one big category called major neurocognitive disorder. There's also minor neurocognitive disorder. I'm not going to get into that. We're just going to focus on the major neurocognitive disorder. So it's even more complicated than this. But essentially, you have to have a significant decline. And here's the key, from a previous level of performance. And so that's often difficult to define, right? Because people typically don't have a baseline measurement of their memory. They don't have a baseline measurement of their intellectual ability, of their executive function or attention, right? So when they go in and they say, my memory's not as good as it used to be, or I'm having a hard time with attention, I'm not able to think as clearly, they'll get tested, and maybe their test comes out above normal. And so the doctor's in there and go, well, you're testing above normal. And so they have to wait and come back in and get another test to show even more decline, where they can actually have a comparative test before often they're given a diagnosis. And it could be that that person started 25% above normal, and they've already lost 25% of their capability because they're just now normal now. So not having that baseline measurement can be difficult, right? And I don't think I'll have time, well, I won't have time today. I think I'll just ask you, there's a 6-minute video on the Moodle site. It's an interview with a physician, and he's talking about this process of how he knew that he was having cognitive decline, but because he started at a higher functioning level than most of the population, he wasn't able to get a diagnosis, and then that affects your ability to get into clinical trials, get medications, and there's all this very complex problem. We'll talk a little bit about that with Alzheimer's disease, and the difficulty with trying to have early diagnosis where treatment is still more viable. All right, so there also has to be concern of the individual or another person that there has been this decline, so it can't just be self-report. There has to be some objectivity to it, and that's where it gets into problems. And then again, the neuropsychological testing documentations, and that it interferes with their everyday life activities. And that's another hallmark of being diagnosed with abnormal versus normal, right? Because this doctor could present and the guy says, well, yeah, but you're getting older. Senile dementia is normal. You're going to lose some cognitive function. Is it interfering with your ability to do your daily tasks? Well, no, not really, but I know I'm having this decline. So again, that becomes sort of problematic. But we have to set a bar somewhere to say, okay, this is normal, and you've now progressed over here, and this is abnormal, we need to diagnose you. All right, so within those neurocognitive disorders, there's a whole bunch of subsets, right? Alzheimer's disease being one of those, but a lot of other things on here as well. So in looking at the causes of dementia, we can separate them into two categories. Degenerative causes that are going to continue to get worse, that are going to be progressive, and non-degenerative, meaning that events are happening like head trauma or stroke that we already talked about, where we're getting discrete problems with brain waves. All right. So in terms of prevalence, Alzheimer's disease is about 70% of dementia. That said, Alzheimer's disease is also kind of the default. So if you're older, you present with advanced memory loss, sort of the default is going to be, okay, you probably have Alzheimer's disease. And we've gotten better about trying to discern if it's something different, like Pick's disease, and I'll talk about, or normal pressure hydrocephalus. It sometimes gets confused with Alzheimer's disease. But this number may be slightly inflated because of that. And then 15% is vascular. We already talked about strokes in the very beginning of the course. But the two of those then account for 85% of dementia in the elderly. And all the rest of this is 15%, including Parkinson's disease, substance abuse, head trauma, other neurodegenerative types of infectious disease, things like that. So we'll spend most of our time focused on those two, but I do want to talk a little bit about how can you discriminate between the types of dementias. And so the first thing that helps you discriminate between the types of dementias is having a good history of what that individual's been exposed to, what their life has been like, and a familial history, because we know that, for instance, Alzheimer's disease has a strong genetic component to it, as does addiction. And so if we're looking at substance-induced dementia, then family history can be important there, right? So we're interested in heredity, we're interested in exposure to drug use, we're interested in travel. Could you have been somewhere and got exposed to a virus? I mean, I think we can now throw in, were you infected with COVID? We don't know what the long-term effects are, if there's going to be long-term central nervous system effects on this. Biotraumatic brain injury, have you been in car accidents? Did you play a contact sport? Those sorts of things. So all of that is very important in trying to identify, where is this originating from? And then secondly, the next important thing is the timeline. And this is where we want to try and discern, is it neurodegenerative or is it non-degenerative? Is it happening progressively, gradually, or is it happening in a stepwise progression, or is it happening in relationship to particular events, right? And so vascular events, the onset is quick, it's sudden, it tends to be stepwise, meaning you have a loss and then you're sort of plateaued and don't continue to have a loss, versus neurodegenerative, which is going to be progressive and not slow down or stop. It's going to be more gradual than the vascular. And then finally, you'd think symptoms would be the first thing that you look at. But really, symptoms are sort of the final thing to kind of try and tease out. Okay, we know it's degenerative. Let's see, what are the symptoms? Is memory the first loss, or is it emotion and personality? That can help us tell us this originating in the frontal lobe or originating in the temporal lobe. Right? So in Pick's disease, that originates in the frontal lobe. Normal pressure hydrocephalus has that symptom of incontinence that's unusual in these other, especially early onset cases of Alzheimer's and Pick's. And then Alzheimer's disease starts typically in the hippocampus with spatial memory and newly formed sort of long-term memories. Where did I park my car? Where did I leave the keys? Those sorts of things. And then, diagnostic tests are really largely experimental at this point. We're getting closer to having tests for Alzheimer's disease, but we're not quite there yet. Brain scans are not quite good enough to detect the types, the etiology. We can often see the damage that's occurred in the skin, but we can't tell what caused the damage. All right, so a little bit about Pick's disease. Pick's disease happens, it mimics early onset Alzheimer's because it happens earlier than most Alzheimer's, but there is early onset Alzheimer's, particularly genetic variants of Alzheimer's disease tend to have an earlier onset. The etiology of it is characterized by degeneration in the frontal lobe first, and then it progressively moves to the other areas of the brain, including the temporal lobe. Not as much in the parietal lobe, but definitely a frontal to temporal lobe progression. So the symptoms then are changes in executive function, decision-making, personality and mood and emotion that are happening as opposed to forgetfulness. And that's sort of, again, when you get to, okay, this is neurodegenerative, how can I define it? What are the behavioral symptoms, particularly the earliest symptoms, before it progresses to affect too much of the brain? So Pick's disease consists of aggregates of proteins, of tau proteins. So tau is the protein that makes up microtubules in neurons, the sort of highways that are going through the axons. And in Pick's disease, these tau proteins form spherical, so they're like bubble-shaped aggregations of them. And this triggers an immune response, and so there's a proliferation of astrocytes, and then there's a loss of neurons. And the brain, at autopsy and histology, has a sponge-like appearance, like where these cellular aggregates were become sort of voids, and the neurons that are there are gone, and there's these little holes, tiny little bubbles throughout the brain. So as I mentioned, discriminated from Alzheimer's disease based on social behavior and personality, emotion as the first symptoms as opposed to memory. There's often social disinhibition, where people say inappropriate things or do inappropriate things because they lose that ability to inhibit unwanted behaviors. They can also have a loss of motivation or drive. And as I mentioned, parietal lobe functions are affected in Alzheimer's disease, but not as much in Pick's disease. All right. There's no treatment for Pick's disease right now, and so that's a fatal condition. Korsakoff's dementia affects a small percent. I just want to mention it because it's a special type of dementia associated with chronic alcohol use. And the characteristics are, it's largely a memory issue. Recognition memory is poor. It's not just, often with Alzheimer's disease, especially in the early stages, people have a hard time consolidating memories, but can still recognize or recall memories. Whereas with Korsakoff syndrome, it's sort of more of a degeneration of the neural networks as opposed to the hippocampus first, more a degeneration of the cortex, cortical areas. They often lose memories that are older, right, so more susceptible to degradation first as opposed to Alzheimer's disease, where they're sort of not having new memories being formed. And the important key fact with Korsakoff's is it happens as a result of chronic alcohol use and poor nutrition. And so what we think happens is that the chronic presence of alcohol is producing little sort of micro strokes in the brain, because alcoholics often get a lot of their calories from the alcohol itself, and so their blood sugar levels can drop and blood glucose levels can be low. And alcohol itself inhibits vitamin B1 binding, and vitamin B1 is needed for glucose transport across the blood-brain barrier. And so we see, here you see a brain scan of a normal brain, of a middle state, and then a late state or end state of Korsakoff's. And you can see that in the early parts, there's a widening of the ventricles, which would be hard to tell if we didn't have a baseline, right? I mean, it's hard to see that these are big. It's obvious here that those are huge ventricles, right? And that there's a lot of enhancement of the sulci and shrinking of the gyri and the cortex. But early on, if we don't have a baseline, it's very difficult to compare that to normal. Not all alcoholics experience this. It's a fairly rare form. The majority of alcoholics don't have Korsakoff syndrome. It has to be paired with that sort of poor nutrition and a really heavy, continuous presence of alcohol to cause that vitamin B1 deficiency. Our body's really good at preventing deficiencies of nutrients, and so you really have to suppress that in order for it to happen. And so this is a nucleus that's in the frontal lobe, and it's sort of a hallmark. Maybe it uses glucose in a different way, or has more need for glucose and is more susceptible to these micro strokes. But we definitely see a selective reduction in that nucleus compared to chronic alcoholics. And then, as I mentioned, an enlargement of the other ventricles and sulci in the brain. General sort of neural loss. Just going to run through some, you can have dementias due to infections. So anytime there's swelling in the brain, that can cause a loss of neurons. And anytime we have a loss of neurons, that can sort of catch up with us, right? So you can have, if you're normal, we're saying that this is normal loss across lifetime, and it's not until we get here that we see senile dementia, like normal dementia. We get to this percentage point, right? But if you're starting out normal, and then there's a drop, and then you continue on that same trajectory, right? We're going to see an enhancement of when that dementia takes place. So if there was a viral event that caused loss of neurons in the brain, now instead of being at 88%, we're at 78%, and we're starting that, we maintain that trajectory. Same thing with drug use. If there's drug use that causes neuronal loss, you can see where sometimes you don't see the behavioral effects until much, much later in life. Right? You don't see the symptoms too much later. In terms of viral exposure, the herpes virus, which we now have medication that can help suppress and control that, so this isn't as big a deal as it used to be, but can cause inflammation, which causes loss of neurons, particularly in the frontal lobe. Prion infections, we don't have any cure for. That's what's called mad cow disease or Creutzfeldt-Jakob disease, where the prions themselves kind of act like an infectious agent, where when they become this shape and they cause other normal prions to also become this shape, and then kind of spread throughout the brain. That's a fatal disorder pretty quick, within only 2 years. Hard to contract it. You have to consume infected prions in order to have this become expressed. So through good food handling, we're able to limit that transmission. HIV can also cause dementia, although we have got, the medications for HIV have gotten so good, the types of therapies that we have, the ability to suppress the symptoms of HIV are so good that we're not really seeing dementia from HIV unless it's left untreated. All right, and then vascular dementia, we already talked about this when we talked about cardiovascular stroke, right? But often, particularly in the elderly, they can begin to have what we used to call multi-infarct dementia, which is where you have these like microstrokes, these little strokes that don't maybe produce, it's not like they're getting paralyzed or loss of sensation or aphasia or some big symptom like that. But it's affecting small, sort of localized areas in the brain. And they kind of add up over time, right? And so the symptoms typically appear as types of amnesia. And again, treatment involves trying to treat the cause. So if it's high blood pressure, trying to decrease the blood pressure. If it's high cholesterol or cholesterol problems, trying to eliminate those things, right? And so this is sort of the type of progression where there might be a deficit, and then there might even be some improvement, and then they have another little incident, and this can happen sporadically over weeks. I mean, it doesn't have to happen close in time necessarily, as opposed to neurodegenerative disease, where you're just going to see that consistent slide with something like Alzheimer's disease. All right, normal pressure hydrocephalus, I think we already talked about in here some, if not. There's not a whole lot to talk about other than the fact that we get increased ventricular pressure from the cerebrospinal fluid that acts on all the structures near ventricles, as well as the surrounding cortex. So the surrounding cortex, the pressure in on the brain is going to produce dementia, memory, and cognitive function deficits. The pressure on the basal ganglia in the lateral ventricles is going to produce movement issues similar to Parkinson's disease, shuffling of gait, that sort of thing. But the pressure in the third ventricle is going to produce incontinence, urinary incontinence. And that's sort of the key thing that we don't see. So often it's misdiagnosed as Alzheimer's or Parkinson's, but this incontinence can be a key. And you can see the enlargement of the ventricles through MRIs, and by putting a shunt in, you can eliminate that pressure and people really go back to normal function. All right, and finally we have Alzheimer's disease, okay? And so for Alzheimer's disease, you have the criterion for the neurocognitive disorder, and then we're going to talk about insidious onset and gradual progression and impairment. So slow, continuous loss of functioning. It doesn't ever stop or recover. Often we want to have evidence of genetic or family history, right, or genetic testing. And there's clear decline in memory and learning, and that it's steady, gradual, without extended plateaus, and it's not due to anything else. It's always like an exclusion criteria. It's not due to substance abuse, it's not due to head trauma, it's not due to something. All right, so it's very common. About 5 to 10% of people over 65, and when you get to 85, it can get as high as 85, as 50%. It's much higher. Again, cognitive impairment, gradual decline of short-term explicit memory. Sometimes there's language impairments also, depending on how it's progressing because of the proximity of Wernicke's area to the temporal and parietal lobe where it's sort of originating in. We can break Alzheimer's into sort of 3 stages. There's an initial phase that is where the hippocampus is initially being compromised, and so we get problems with things that the hippocampus does. Spatial memory. People can't remember how to get places. And then we have like sort of what I would call short-term, long-term memory, like newly encoded sort of long-term memory. Things that you want to hold for a little period of time and not remember, like where did I park? I want to remember where I parked in this parking lot, but I want to remember that tomorrow when I park in a different parking lot. I want to remember that information. So this sort of short-term consolidation process becomes compromised. The second stage typically occurs 2 to 3 years after the first, and you start to see more progressive memory failure, right? So not just encoding new memories, but having problem retrieving older memories, because now we've moved beyond the hippocampus, and now we're compromising the neural networks that are in the temporal lobe. And that's sort of the progression. There's hippocampus, temporal lobe, parietal lobe, frontal lobe. And then once we get to the frontal lobe, you start to lose all intellectual functioning, and people sort of become in a vegetative state. They'll often stop eating and drinking, and so unless you put in feeding tubes, that's sort of an end-of-life thing. They'll often develop pneumonia because they can't get out of bed, so fluid builds up in their lungs. So people don't die from Alzheimer's disease, they die from the complications of this vegetative state that they're in. And I'll talk more towards the end or on Wednesday, but there's this big sort of preclinical period where people may start to notice issues, but there has to be like a significant decline from normal aging to get actually diagnosed with a mild cognitive disorder. And then you're not really have the serious symptoms of dementia yet, and so you're in this mild cognitive impairment area before you get to an actual dementia sort of diagnosis. All right, so what's happening? There's genetic components for sure. If a first-degree relative has Alzheimer's disease, your risk increases. It's related to chromosome 21. The precursor protein for the amyloid beta protein is encoded by chromosome 21. Because clients with Down syndrome, people with Down syndrome have 3 copies of chromosome 21, they overproduce amyloid beta precursor protein, and therefore the rates of it being miscleaved are much higher. And so they tend to develop early onset Alzheimer's disease. It probably involves more than one gene. We know chromosome 21 is important. We also know that there's this APOE gene that is also involved, this single nucleotide polymorphism. And if you have two variants of this copy, then then the chance of Alzheimer's disease is very high, 91%. It's not 100%, so 10% of the population has this variant but does not develop Alzheimer's disease. So it's not a definitive thing, but it definitely is involved in this system, right? And if you have no copies, still 20% of the population with no copies of this develop Alzheimer's disease from other reasons. So again, it's complicated. There are definitely genetic things involved with this, and therefore that represents a potential treatment with gene therapy in the future that we can go in and change this variant early on in life, potentially. So we haven't quite got there with human gene therapy. All right, there's also some evidence that exposure, so lots of different things might contribute to the formation of Alzheimer's disease. So exposure to particular environmental toxins, particularly aluminum, it seems like. Exposure to aluminum tends to create neurofibrillary tangles that we see with Alzheimer's disease. Disrupted metabolism. There's an increased risk of Alzheimer's disease in people with diabetes, particularly type 2 diabetes. And also we'll see in mice at least that exercise and having a strong cardiovascular system and good glucose metabolism can actually decrease the risk of Alzheimer's disease. So availability of glucose and metabolism plays a role in this as well. The olfactory system is affected by Alzheimer's disease, and so one of the early deficits that we can, that tests that could possibly be used for diagnostic purposes is a disruption in the smell ability. If that is difficult because it's not anosmia, it's not like lack of smell, so they don't have any smell, but it's a decrease in discrimination and perception. And one, again, if you don't have baseline measurements, it's hard to tell if there's a change. Two, administering olfactory tests that are sensitive enough for discrimination and threshold sort of determination are difficult to do. Particularly if you're not in a laboratory like research setting, if you were in like an ER or something like that, it's going to be difficult to do.
Is there any suggestions for people who do have Alzheimer's in their family to like document their baseline symptoms that if they start getting tested?
Yeah, I mean, I don't think that there's like formal recommendations to that, but certainly you can get genetic testing done. And that's sort of the debate about, do you want to know if you're predisposed to this? If there's really nothing you can do about it, right? Or maybe there is, I don't know, but that's an individual sort of ethical choice for individuals. And then, yeah, so having baseline tests done could help with that, right? Could help identify, particularly if you have a feeling, I mean, it's the same thing with people that have a history of breast cancer that might want to get a preventative mastectomy or something like that. I mean, there's preventative things that people can do if they're concerned about that. So if there's a strong history of Alzheimer's disease in a family, maybe you would want to be trying to get into clinical trials and wanted to be able to catch it as early as possible if you think there's in the population that's susceptible. All right, so we know that there's losses of synapses. I'll talk about the relationship with acetylcholine. There's particular loss of acetylcholine receptors. Acetylcholine being sort of that arousal state in the brain that keeps us like wakeful and alert. Not hyper-alert norepinephrine, but sort of the general awake. And so as this disease progresses, part of the decline of cognitive function is the brain's just not as alert. It's almost like it's sleeping. Right? All right, the two principal histological things, if we go in and we look in a brain post-mortem, we see neurofibrillary tangles, which are bundles of abnormal tau filaments. Right here we see them. They form like a helix shape around each, they like wrap around each other. and form this tangle, which is different than the tau tangles in Pick's disease. The tau in Pick's disease didn't tangle around itself, it aggregates in those spherical sort of balls. And these tend to happen in large pyramidal neurons, because the pyramidal neurons have very long axons. Those are the neurons in the hippocampus that are reaching out into the temporal lobe to form neural networks. They're the neurons that are crossing through the corpus callosum. So those are all, they have, you think, okay, well, if you have a really long axon, you're going to be making a lot more tau than if you have a short axon. You're not going to be making so much tau, there's not so much opportunity made for that tau to form these misshapen neurons. So motor neurons, hippocampus neurons are commonly affected. Amyloid beta plaques is the other sort of major histological thing that we see. And so here, the amyloid beta protein gets cleaved inappropriately with 42 amino acids instead of 40. That makes it useless, and it aggregates in these clusters, and we think these clusters are neurotoxic. They interfere with synaptic signaling. I'll talk about their effects on particularly glutamate and acetylcholine in a second. And there's an immune system response to them. So we have microglial astrocytes that come in and can cause local inflammation by releasing their sort of normal immune response. They don't recognize what is this thing that's happening. We need to go in and treat it like it's a foreign antigen. And so part of the decline in the brain could be like a co-expressed immune response that compromises brain cells as well. All right, so these loss of cells, as I mentioned, particularly affect, if we look in the hippocampus, we see large pyramidal cells that are affected throughout, and then particularly in this entorhinal cortex, we see a lot of plaque formation early on there. Eventually, this does spread throughout the entire cortex, but it originates in the hippocampus for whatever reason, because those neurons are more susceptible, maybe because they're being used so much. We don't really know, we don't understand. A lot of what we know about Alzheimer's disease are snapshots, right? Somebody thinks that, maybe they think they're in the early stages of Alzheimer's disease and they end up in a car accident. And so they do an autopsy and they look and they find the plaques just in their hippocampus and it hasn't spread to the rest of the brain. So these are the types of snapshots of evidence that we have, and we're trying to just piece it all together, but it's really just these kind of images of post-mortem brains that we're trying to discern all this from. All right. The last thing I'll just talk about, and I'll save the rest of these for Wednesday, we'll have time to go over them Wednesday, is the creation of this amyloid beta 42. And so there's this amyloid precursor protein. It's encoded by the 21st chromosome. And it gets cleaved by some enzymes, alpha secretase, beta secretase, and then this gamma secretase, which results in a protein that should be 40 amino acids long. And normally, this exists in very low levels in our brain. We don't really know what its function is, but some people think that it has a neurotropic function. It helps keep the blood barrier, the blood-brain barrier strong. It has some sort of impact on that, and then it helps maintain calcium stability. And I think a lot of this is kind of when we disrupt this normal system. One piece of evidence is that the blood-brain barrier looks weaker in Alzheimer's patients, and we have calcium instability there. So again, it's sort of difficult. We don't really know the normal function of amyloid beta 42. But when we have low levels, this is sort of the normal process over here. And when we have higher levels, when we start to get amyloid beta 42 involved, and that can be negative as well. So I'm going to stop here. I'll pick back up here Wednesday, and then we'll talk about a little bit about why psychological stress is stressful, and then we're going to talk more about that towards the end of the course. A little bit about what makes humans unique compared to other animals.